Hi guys, I'm sorry for the continued interruption. Let me move this out of the way a little. All right, let's try to finish this up. This is a long lecture, I know, so maybe it's good that it's in pieces. Um, so we were talking in the previous lecture about how cells can make reactions spontaneous. So if there is a small positive delta G, using a biochemical pathway, they can change concentrations. They can increase the concentration of the reactants. They can decrease the concentration of the products and push the reaction in the forward direction. But we said that if there was a large positive delta G, a large requirement for free energy, that they would have to couple that to a different process or a different reaction that produced enough energy to power it. And that oftentimes when that happens, such as in the case of ATP hydrolysis, that's going to be a coupled process where you create an intermediate molecule like this charged B, this B phosphate. So it's not really A plus B goes to AB, it's really A plus B phosphate becomes AB. All right, and because of those equilibrium, what do I want to say? Not equilibrium effects, because of the concentration effects, the cell does not want to be at equilibrium, right? Because at equilibrium, there's no capacity for work. At equilibrium, delta G equals 0. There's no capacity for work. And so cells have lots and lots of work to do. And so instead of being at equilibrium, cells maintain what's known as a steady state. They keep certain molecules at specific concentrations, such as we talked about, ATP concentrations are kept very high, while ADP concentrations are kept very low. Similarly, the ionic balance, the for osmoregulation, such as the concentration of protons, it's not kept at equilibrium. Those concentrations are very actively maintained, and in fact, maintaining the proper ionic balance accounts for almost 1/3 of the ATP that is produced by metabolism. It is a very, very important process, not just for keeping the water balance of the cell, but also for powering other processes by secondary active transport. So, what kind of cells will maintain equilibrium? Only dead cells. That's very dramatic, isn't it? Okay. So think about this one for a minute. I just told you the answer. Right? This is because this cell keeps the ATP concentrations high and the ADP concentrations low. All right, so let's shift gears a little bit because enzymes have a lot to do with energy and talk about enzymes. Now, if you feel like you don't have a very good grip on enzymatic reactions, I highly encourage you to go back to your introductory biology textbook and read the section on enzymes because we're going to talk a lot about enzymes this semester. And so you should really have a good grip on an enzymatic reaction. So, well, before I get ahead of myself, let's talk about some of these definitions. Enzymes are proteins, and they catalyze biological reactions. Please excuse me. So if you think about a chemical reaction, there are many, many chemical reactions that may or may not take place spontaneously. Just because a reaction is thermodynamically favorable does not mean it's kinetically favorable. Okay, I know that sounds kind of strange, but thermodynamically favorable means that the overall delta G is negative. But you have to get those molecules together in the right orientation in order for them to react. And so sometimes that is not a favorable process. It's a function of probability, right? So while the reaction of glucose becoming carbon dioxide and water is favorable thermodynamically, right? It's a spontaneous process. In fact, that's the whole basis of glycolysis and aerobic respiration. We're getting carbon dioxide and water from glucose, and we get energy out of that, right? But your paper is made out of glucose, right? It's made out of cellulose, which is made out of glucose. You are not afraid that your paper is going to spontaneously combust and become carbon dioxide and water and a flaming pile of energy on your desk, because those reactions are not kinetically favorable. You have to have a catalyst, right? So what's one way that you could make your paper turn into carbon dioxide and water? Right, you could light it on fire. You could take a match or a cigarette lighter and provide the activation energy. You just get the process started, and even though you had to provide a little bit of fire in the form of a match, you get a lot more fire than you started with, right? But a cell can't light itself on fire or it's dead. A cell can't shake itself up. A cell can't change its pH. And so a cell uses enzymes. Enzymes are biological catalysts that are really quite amazing because they can do these chemical reactions that require high heat or high pressure or radical changes in pH in the lab, and they do it all under biological conditions. Some enzymes require prosthetic groups. Okay, actually many enzymes require prosthetic groups. Prosthetic groups can be pretty powerful when they're combined with a protein. And if a prosthetic group is in an enzyme, it is known as a cofactor. Okay, so prosthetic groups of an enzyme are called cofactors. If the cofactor is an organic molecule, it is known as a coenzyme. Okay, so remember from last week, we talked about prosthetic groups, and we talked about heme, and we said, well, heme has iron in it, so it's not organic. That's a cofactor. Okay. We also talked about retinol, which was that lipid that's required for rhodopsin to sense light, because when retinol was stimulated by light, it changed a cis bond to a trans bond. Retinol is a coenzyme because it's a lipid. A lipid is organic. Retinol is also a cofactor. Okay, so all cofactors are also prosthetic groups. All coenzymes are also cofactors. So it's like a nested system. All right, this is something you should be very familiar with. And so I'm going to fly through it. An enzymatic reaction consists of a substrate. The substrate is the reactant of the reaction. The substrate binds to the enzyme in its active site. The enzyme facilitates the reaction somehow. The substrate turns into the products, and they are released by the enzyme so that the enzyme can go find another product. Another substrate. Okay, the enzyme is not changed in any way by the chemical reaction. Most of the time, substrates will bind to the enzyme in a non-covalent way. And this makes sense, right? Because you have to get the products out of the enzyme. In some cases, there are covalent interactions, and in that case, usually the covalent bond has to do with the reaction mechanism. And we'll look at one of those reactions in just a little bit. All right, you should be able to reproduce this energy diagram, okay? An energy diagram shows you the course of the reaction. You have G, right? This is G, not delta G, just G, free energy. Gibbs free energy is on the y-axis, and time, or the course of the reaction, is on the x-axis. So the free energy of each of the molecules will be plotted. So you have your reactants, and they have this amount of energy, and you have your products, and they have this amount of energy. So we can see here that glucose plus ATP becoming glucose 6-phosphate is an energetically favorable reaction. This is the first reaction in glycolysis. Right, this involves ATP hydrolysis. Of course it's energetically favorable. But it requires an activation energy. And that's because in order for glucose and ATP to react, they need to form a transition state. Reaching this transition state takes energy. Now, in the course of a favorable reaction, the transition energy will be lower than the overall energy required. But that doesn't mean that it can be achieved, right? So if you think about a boulder sitting on top of a hill, it has a lot of potential energy based on its position. Because if that boulder were to roll down the hill, it could cause a lot of damage, right? It's going to have a lot of motion. But getting the boulder from the top of the hill to beginning its roll down can be a lot of work. And you might not be strong enough to push it off the hill. Okay? So although it's got the potential energy, and if it could get over the lip of the hill, it would have a lot of kinetic energy. Starting the motion, getting through that inertia, is the activation energy. And what an enzyme does is it lowers that initial activation energy requirement. See, there's my boulder sitting on top of the hill. You push that boulder off the top of the hill, right? But if you had a wedge, where you could use a wedge to help lever this boulder off the hill like this, that would be like an enzyme. It's like a tool that you can use to lower the energy requirement. Now, instead of just pushing with my hands, I have this mechanical tool that can help me get the boulder off the hill. Okay, this is analogous to how enzymes work to lower the activation energy. All right, so this is modeling the transition states. The activation energy is the requirement to get the molecules into their necessary transition states so that they can be pushed to the other side. There's three main mechanisms of catalysis. Now, I want you guys to appreciate the fact that you could take an entire semester just on enzyme catalysis, okay? So these are three very general mechanisms, and there are many, many, many more. Each enzyme is unique into itself. And, I mean, enzymes are just amazing little things. But these are three general mechanisms, okay? The first one is to lower the entropy of the substrates. Okay, so if you think about the cytoplasm of the cell, is a very crowded place. And in order for two molecules to react, they have to come together in the proper orientation. If there's no enzyme to facilitate that, it's all just Brownian motion. The molecules are jumping around and bopping around. If they come together, they might react. They have to come together in the proper orientation. So it's a function of probability. And so in this case, the enzyme is lowering that probability by attracting the substrates into the active site, and they enter the active site in a very specific way. So just putting these two molecules into the active site so that they come together properly, you facilitated the reaction. Okay? You're lowering the, or I'm sorry, you're increasing the probability because you're attracting those substrates. Okay, you can also actually increase the reactivity of those molecules, and this often has to do with stabilizing that transition state. You're making them more likely to react. And this can happen in many different ways. Oftentimes, you'll have either acidic or basic R groups inside the active site um, that make it more likely to react by donating or accepting electrons. So it actually is charging it, making it more reactive. Polar R groups can do that, although it wouldn't be as strong. You can hydrolyze things using water. Cofactors, oftentimes ions, ionic metals, will be inside the active sites, and that will help to make those substrates more reactive. Okay, so changing the chemistry of the environment. Okay, this is what increasing reactivity means, is that you're making a small microenvironment that's actually changing the chemistry that those substrates are sitting in. Okay, and then the final way, well, not the final, but the final one we're going to talk about is an actual physical stress to the molecule. And this is what's often seen many times. The induced fit model is often talked about with enzymes, and that's like the substrate enters the active site, and then the enzyme kind of changes its shape to more closely fit the substrate, kind of like a glove sliding onto a hand. When this induced fit model occurs, it often involves stressing the bonds, a physical stress to the bonds of the substrate. And that's going to increase the likelihood of reaching the transition state and moving to the products. All right, enzyme inhibition. There's two different types of enzyme inhibition. Reversible inhibition is the type that cells want to engage in because this is the way that they can turn enzymes on and turn them off so that they can regulate exactly which biochemical pathway is going to occur today. On the other hand, toxins will often capitalize on enzymatic reactions, and they will bind irreversibly to the enzyme, maybe in the active site, maybe just to change its conformation, and then they're covalently bound and they don't let go. And sometimes these are synthetic chemicals, sometimes these are produced by biological organisms to poison their predators, or in the case of plants, they're herbivores. So irreversible inhibition will poison the cells, right? Because it's difficult to make an enzyme. It takes some time. If you kill all the enzymes and the plant can't make more, or the cell can't make more, then it's dead. Maybe if it's a required biochemical pathway. We're only going to talk about reversible inhibition. And we're going to talk about competitive inhibition and non-competitive inhibition. And both of these inhibition types are used by all types of cells. And sometimes the same enzyme may be acted upon by both competitive inhibitors as well as non-competitive inhibitors. So for your follow-up assignment, I would like you to look up the graphs of these enzymatic reactions with competitive inhibitors and non-competitive inhibitors and Vmax. It's in your book and it's on the internet. And so I want you to draw those um, um, reaction graphs and describe for each graph for competitive inhibition and non-competitive inhibition. I want you to describe why Vmax is reached when it's reached. Okay, so for competitive inhibition, you have inhibitors that are similar chemically and physically to the substrate, and they're capable of inserting themselves into the active site. And so it's literally a competition for the active site. Is the inhibitor going to enter the active site, or is the substrate going to enter the active site? Because if there's an inhibitor in there, then the substrates can't enter. Okay, but you have to remember that as these are enzymes, these things are constantly popping in and out. Okay, so the inhibitor can't stay inside the active site indefinitely. It goes in, it comes out. It goes in, it comes out. If this system is very dependent on concentration effects. As long as there's some substrate in the environment, there will be a low level of reaction. So it's very much concentration dependent. If you have a higher concentration of inhibitor to the substrate, then your inhibition will be high. Okay? If your inhibitor concentration to substrate is low, then you'll have much more of your enzymatic reaction occurring. Okay, but you'll still have a good bit of enzymatic reaction occurring. Well, maybe not a good bit, but you'll have some low level of enzymatic reaction occurring even in the presence of inhibitor. Now, there's several different types of non-competitive inhibition. Covalent modifications will shut the enzyme on or off, and these are things like phosphorylation. Okay, so if you phosphorylate the enzyme, then it changes its shape and it's no longer capable of acting on the substrate. Or it could be the opposite. If you phosphorylate the enzyme, you might turn it on. Okay, so this is a very powerful way that, and a very common way, that the cell regulates its enzymes, phosphorylation. But there's also allosteric regulation, and this is also non-competitive because it doesn't act in the active site, it acts at an allosteric site. But this isn't covalent. There's no covalent bonding here. Okay, this is more like the way that the substrate interacts with the active site. And there's also two different types of allosteric modulation. So these both happen. So different enzymes work in different ways. Allosteric modulation, allosteric inhibition is much more, the amount of inhibition really depends on the concentration of the inhibitor. Okay, because it's not a competition. If the inhibitor is there, it will inhibit the enzyme. Okay, there's no competition for the active site because the allosteric inhibitor actually changes the conformation of the active site so that the substrate's no longer capable of binding. Okay, so first we're going to talk about this scheme on the top here. And this can be either turning on or turning off the enzyme. Okay, so in this case, the allosteric regulator is turning on the enzyme. And in this case, the allosteric regulator is turning off the enzyme. So this is the allosteric inhibitor. This is the allosteric activator. Okay, you have your active site where the substrate wants to bind. In the case of allosteric activation, if there's no activator in the allosteric site, then the substrate is not capable of binding. Okay, the active site is chemically and or physically incapable of accepting the substrate. When the allosteric activator enters the allosteric site, it causes a conformation change, and now the substrate fits into the active site and the enzyme is turned on. Okay? The same thing can happen by an inhibitor, but in this case, if there is nothing in the allosteric site, then the enzyme is on, so the substrate can bind. When the inhibitor enters the allosteric site, it changes the conformation of the active site so that the substrate can no longer bind. Okay, so that's on or off, right? If the allosteric activator is in there, the enzyme is on. If the allosteric activator is not in there, the enzyme is off. There's also what's known as allosteric modulation, and that's what you see here. These kinds of enzymes, with no regulators in them at all, are constantly in flux. Okay? So usually they have some sort of a probability. They'll be on 70% of the time and off 30% of the time. Or they'll be on 10% of the time and off 90% of the time. That's innate to the enzyme. And different enzymes will have different probabilities of being on or off without any regulators. So they're constantly flip-flopping forms. On, off, on, off, on, off, on, off. And then an activator comes and it binds into an allosteric site and it stabilizes the on position. It also usually will cause other allosteric sites to be more likely to accept additional allosteric activators. So usually once one allosteric activator binds, it causes the binding of other activators. Okay? So now the on form is stable, and it's not going to flip-flop so much. It can still flip-flop, but it's less likely. You've altered the probability of the change. Okay? On the other hand, if you're flip-flopping, there's no regulator, on, off, on, off, on, off, on, off, and then an inhibitor comes, and it's going to bind in an allosteric site, and it's going to stabilize the inactive form. And sometimes, often, maybe, it will also stabilize or increase the probability of more allosteric inhibitors binding. Okay, so in the top mechanism, the enzyme is either on or off. And that's going to be based on the presence or absence of an allosteric activator or an allosteric inhibitor. In this bottom mechanism, these enzymes flip-flop between the on and off form, and the activators and inhibitors are going to stabilize one form. Okay, these both happen. This is not, I'm not sure which mechanism is true. This is some enzymes do this and other enzymes do that. Okay? All right, an enzyme in a metabolic pathway is regulated by a competitive inhibitor. Competitive. As more substrate is produced, what will happen? Okay. Competitive inhibitors are very dependent on the ratio of substrate to inhibitor. So if you produce more substrate will be converted to product. Well, what about for the allosteric? An enzyme is regulated by an allosteric inhibitor, and it's being inhibited. If more substrate is produced, what's going to happen? Because allosteric inhibition is not dependent on the relative ratios, or not very dependent on the relative ratios of substrate to inhibitor, even though more substrate is being produced, you'll still maintain the same level of inhibition. Sorry, let me just... Okay, I think I'm going to stop there. I think your brains are mushy enough. And next week we will talk about metabolism and high energy molecules and redox. So please go over those. Um, and hopefully we'll have time for some membranes also. So for next week, I want you to, what did I tell you to do? I'm going to tell you to do an assignment on pollen. That's my other class. I'm going to write out what you're supposed to do on the internet. Oh, yeah, you're supposed to do the Michaelis-Menten things. The Vmax, like the graphs with the enzymes. I'll write that up underneath the videos so you know what to do. We have a quiz next week. And also, make sure you watch the TED Talk that I've posted below. Oh, baby. Who's my baby? Yeah, but first I have to post these for my students. And I'll let you watch.